Okay. Okay.
All right. Good morning, everyone, students, families, and colleagues. I'm Olivier Gauthier, professor at the University of in France. I will be moderating this first session. This will be a session where 3 of our students will be presenting their thesis. Joining me in the jury are Martina Mokarik, a researcher from Cornell University, Estelle Blanco, assistant professor from the University of the Basque Country, and Nico Whiteberg, the assistant professor from the University of Oviedo. So, as you know, students, you have 15 minutes to present your research, then we'll have a 15-minute discussion. You will be then taking questions and discussion with the jury primarily. If time allows, we will allow the public and the people watching and following us online to also ask questions and make comments and congratulate our students, of course. I will be waving my hand when we come to the 2 minutes left in the presentation. I'll just to warn you that it's going to be time to wrap it. Okay. If you see me standing, it's that you're mostly over time.
Oh, okay. Yeah. I'm gonna take my time, don't worry.
All right.
Okay.
Thank you very much.
Okay, so good morning, everyone. My name is Aliyah, and thank you for being here for my presentation. I'm so excited to share with you my master's thesis project, which is talking about the green juices as an alternative media for sustainable microalgal protein production for aquafeed, which took place in a bioresources recovery lab at Ghent University under supervision of Dr. Marcela Fernandez de Souza. So, a quick overview about the aquaculture. So, as we know that aquaculture is a fast-growing sector for food production, and it's crucial for to meet the global demand and also to ensure the food production. So, as we can see here for the FAO report 2019, we can see that the aquaculture has been increasing since 2019, and it continues growing for the global demand, and also agriculture relies on the fish meal as a primary protein source of the aquafeed. So why it is? First, because fish meal is a high quality protein, so we can see that it's almost more than 60 or 65% of the protein content, and also it's packed with essential lipids, omega-3s, and also it enhances the performance and the breathability of the fish, which increase the feed intake. And finally, according also to the FAO report 2018, so more than 70% of the fish meal has been used in agriculture. So we also still have downsides and major concerns. First is overfishing of the wild caught stock fish, and also the high prices due to the global demand, and finally the higher environmental impact. So to solve this problem, we found a solution that microalgae can be a strong candidate for that, and we choose Nanocorapsis oceanica to be as an alternative protein source for fish feed. Why? First, because it's a marine microalgae and it's backed with high protein content between 40 to 50%, and also omega-3s and fatty acids. And also it contributes in different biotechnological application, which is animal feed, fish feed, nutraceuticals, and also biofuels. And to continue the growth of it, so it doesn't require urban land to grow in, and also it doesn't require the fresh water to grow in. And it's fast growing because it can be grown in a small scale in the lab and also in a larger scale in the industry in open pots. But however, we have cons, which is the cultivation of the microalgae is still costly because using the synthetic and normal media for it, and also it contributes in the environmental footprint. So my goal to keep the pros and to try to reduce the cons by changing the media that we are using in it, which is the green juices as a normal media. So before digging deeper into the project, so I would like to talk a little bit about the broader context of the project, which is my thesis is a part of it. So Reduce project is a learning project in Ghent University, and the goal of it to reuse the agriculture waste stream into added value products. And we have three different goals. The first one to reduce agriculture waste and to use the juices as a medium, that the one that I'm working with, and also to support the circular bioeconomy. So first, that we are collecting the juices. So we collected the flower juice, leek, and chicory, and we are mechanically pressing them into two different fractions. So the first fraction that we have, it's a liquid fraction, and this is the juices that I got from them, and the solid and fiber fractions. These two, and also we have two projects that other labs working in it. One, we are using this in two different applications. So my objective, the first one, that can green juices replace commercial fertilizers for cultivating the microalgae or not? And if yes, so does it influence the protein production and efficiency, as we can use it as a sustainable alternative to fish meal in aquafeed also or not? So for the experimental setup, so first we adjusted the light, temperature, and we had orbital shaking, and also we adjusted the pH at 7. And once we cultivated, so we cultivated the media for 7 days, and we monitored to accounting the algae by using new better and improved chamber, and also we did the data analysis by using Excel. So for the salt optimization, because it's marine microalgae, so of course it needs salt. So we did salt optimization we tested 5 different salt concentrations, and we found out that the highest biomass that we had, it was at 24 gram per liter, which means that the Nanocorapsis sushanka thrive in a highly salty environment. And then once we got the confirmation from the salt, so we did a characterization of the green juices. So first we measured the pH and the electric conductivity, and we got the instant results from it. And then we did the nitrogen content by using the exam method to measure the nitrogen content in each juice that we have. And finally, we did ICB elemental analysis to quantify all the macro and micronutrients inside each juice. So first, we did a screening experiment with the flower juice. So here we had 4 different conditions to evaluate whether the essential elements will be important or not, and if supplementation will be important or not. And we did the inoculum, we started the inoculum at 2 million cells. So for the screening experiment, we had the first one. It was the control artificial seawater as a control media, and then we asked 3 different questions. The first one, that did flower juice the source of everything, and it was yes. And then we had the flower juice source of nitrogen. It also was yes. And finally, the flower juice is source of like everything. It was low. We didn't get any results from it. So once we had the baseline, so we had to like optimize the flower juice as a growth media by we already have the flower juice with everything, and then we had to know that the trace elements and the micronutrients could be beneficial or not, and we can add or remove elements from it to see if the microalgae will grow on it or not. And this time we started with 10 million cells to ensure that we don't have any unwanted substance or any contamination in the culture that we have. So here we had like the two first columns. This one and this one, it was a control. And then we did like, if you have flower juice without trace elements, the algae will know it will grow or not. So we had a mild growth. And then we added flower juice with seawater with only iron. And we can see that the growth is restored, which means that the iron was a limiting factor. And then we did the same, but we removed the calcium from it. And then also we had a low growth. And then for here, we did the same, but we removed the salt. It was no salt in the media. And also we had a low growth. And finally, for the without magnesium, so we had like a drop, like a sharp drop in the growth, which means that also the magnesium and calcium and iron was a limiting factor for the growth of the nanocorruptive channel. So after we get a confirmation between the two media that we have, the first one, the artificial seawater, and then the second one, that the flower juice with everything. So we had to do protein quantification and extraction to evaluate the protein content in each media after cultivating the microalgae. So first we had the microalgae, and we dried in the oven. And then we grained it to get a fine powder. And we added the SDS solution to solubilize the proteins. And then we did incubation for 5 minutes, and then we did centrifugation for 5 minutes to separate the supernatant and the biomass. And then we added 150 ml from the BCAA reagent for the protein bioassay. And then we incubated for 30 minutes to ensure that the reaction is activated, and then we got results from the spectrophotometer. So the first result that we got, it was the protein content. So here we can see that the protein content in artificial seawater is a little bit slightly higher than the flower juice, and also for the dry weight, we can see also the flower juice is slightly higher than the artificial seawater, and this may be because like flower juice has like more nutrients, so that's why it gave like a higher dry weight. And for the extraction, so we did the extraction. So the extraction we tested into 3 different temperatures. Thermal extraction into 3 different pH, the 5, 3, and 9. So for the first temperature that we got, we had a very low protein extraction across all the conditions, especially the flower juice, and which means that the 50% wasn't like sufficient enough to break down the cells, to break down the cell wall of the microalgae Oceanica, and then we leveled up to 70%, and here we got like the results are very good, and yeah. And here for the 85%, so yeah, still we had a good results, but also wasn't as the 70 degrees, which means that maybe the 80% was a little bit harsh for the for the protein extraction, and maybe it did protein denaturation. And also our main focus was on the temperature, but also we had a little bit like different in the pH. After adjusting the pH into 5, 7, and 9, the pH went back into 6. So finally, So finally, among all the three juices that we tested, the flower juice stood out and showed the highest potential. And also supplementation is crucial, especially if it's a trace element, but also flower juice can work and improve most essential nutrients naturally. And also we found out that the thermal extraction at 70% was the optimal temperature that we got. But also for future studies, we need to investigate more the macro and micronutrients role in it and how it can impact the growth of the microalgae, especially in the complex media. And finally, we need to find more ways or extraction methods to extract the protein from it, because we already used the one method. And thank you for your attention, and yeah. Thank you.
For opening this up with control, and you did it with great talent. I want to turn it over to my colleagues and Julie, who wants to start, please.
Great presentation.
Thank you.
I just want to curiosity, since your objective was to reduce the cost of the
Yeah.
using the app now. So, did you try to estimate the cost of this procedure?
Actually, no, because all the juices that we got it was collected before from my lab. So they already have the juices, and also I just did one juice. So actually the cost, no, we didn't do any estimation according to the socio-economic costs, no. Because we still need to work more on the project. I mean, it was a little bit preliminary studies. So if I will continue working with the same project, of course we will have more information about everything.
First of all, congratulations.
Thank you.
I like the work, and also I noticed that you have done it long and hard that work, because you have done it before, and you really see now. So, well, some curiosities regarding your work. Nobody had this money McCarthy. Why did you do a trial to know which solvents increasing?
I'm sorry?
Knowing that the strain that you use is mine, why did you do the trial of salt?
Okay, yeah. So because we didn't know that the salt concentration inside the juices itself, so we had to test five. Yeah, I mean we had to test the 5 different. Wait. Yeah, so we had to test 5 different juices to get at least the optimal salt concentration that we can start working with. Yeah, I mean when I was writing my paper, so I found a lot of papers talking about oh yeah, maybe this kind of microalgae, especially Nanocrops oceanica, they can grow at 30 gram per liters or maybe 40 gram per liter. It's not going behind that, but yeah, I mean for us we already did a triplicate, and we had confirmation that at 24 gram per liter it was the growth was okay, and we didn't want to risk it because it already took us very long time to do it, and once we got a confirmation from that, so we wanted to start with this results. Yeah.
And also, I like the work, but I need statistical analysis. So why you don't do further statistical analysis?
Yeah, I mean because we didn't have any duplicate or replicate, so my supervisor suggested that we don't need to do any statistical analysis, and Excel will be good and will be enough for that to get results from it.
So for experimental only just intermediate.
Yeah.
Finally, I have to say that you have to improve the protein extraction. So for next experiment, which changes
Here?
Yeah, I mean, your conclusions, one of them was that you have to improve the protein extraction.
Yeah, it was we already tested one. It was a thermal extraction. So we also several between 3 different temperatures, and we didn't find like only it was 70 degrees that it was working perfectly well with that. But like when, because like we already found papers that talking about like, oh yeah, maybe during like 85 degrees that we can get better results. But like when we tried this one, it didn't work. And also I suggested that because like we already used like a complex media, the flower juice, so maybe it was really hard to break down all the cell wall of the nanoparticles and extract all the proteins from it. So I like suggested that maybe we can use like a bit milling or like ultrasonication to get the protein extraction. But also my supervisor, we wanted to do that, but also we didn't have time because like the time was like 5 months, and we did the protein extraction like at the end of April, so we didn't have time to do another one. Yeah.
Okay, that's all. Thank you very much.
Thank you so much.
Well,
Thank you so much.
I really like the presentation. Very nicely explained, very well presented.
Thank you so much.
Thank
you.
But yeah, I have the same concern as sisters about the lack of use of statistics.
Yeah, I know.
But so it seems sometimes that you didn't have replicates, and I guess that's the reason why you don't have a standard deviation.
Yeah.
But in some others, I see a standard deviation.
Yeah.
Can you explain why, and in those cases, why did you?
Yeah, because
why
like
did you do a stats?
okay, so for why we didn't do a stats?
Why did you do a stats when you have replicates and standard deviation? Because you have something.
Yeah, I mean like we saw that using Excel that will be convenient for the thesis, and like actually like it was my decision to not do stats. So yeah, I was like discussing with my supervisor, and she saw that if we did not, it's okay. And also for this experiment, we only did duplicate, but for the other one, that's why we had some division because like we did triplicate. Yeah, I mean like. Yeah, maybe if we will continue working on the project, of course we will do that. I mean, like we still have like a lot of stuff to figure it out how we can improve the project.
I guess you're worried that you want to go in and try to publish this result.
Yeah. Yeah.
You need to go through.
Yeah, I know, yeah.
Oh, that's it.
Yeah, thank you so much, thank you.
Any more questions? No? We have time to take some more questions from this. Okay, well, thank you
Really?
very much.
Okay, thank you so much.
I think it's not recorded before, right? Yeah, so the PowerPoint, that's the problem. So this has to be off because it just turned off right after you stopped. This is you, right?
That's me, yeah.
Which one do you
Use
prefer?
the PowerPoint because there's no animation.
Just a second, we're gonna check if it
Yeah,
works.
no, it's way better up here.
We can start with 1938. Start at 1938.
Okay.
Online.
Yeah, true.
So,
Wait, I'm just going to check.
I still have to we will start seeing it on the YouTube. We don't see anything from the video. We see this.
No, then you're not live. Then you're not live, I think.
I am like, yeah,
I can see it.
that thing from the chat, you're not seeing the video at all. Get this done. Okay, it changed. But they were not getting the presentation from anywhere.
Yeah, the one before me.
Ah, no, no, no. So they were seeing the camera, but not the slides.
Okay. We're good now. Understood you.
Okay. Yes. Good luck.
Thanks.
We still are going to wait until 9:30 to start.
I guess they're watching together. It's actually not the worst place, but the.
Listen to it, and then. All right, we move on to our second presentation of this session. So let's look at this work where you work at the linking human activities pressure on ecosystems and bioprocess situations in this setting. So I guess some inter or pluridisciplinary work we're going to be showing us. So Nicholas, you have 15 minutes.
Hi everybody, I'm Nikki, and I will present to you my thesis work, linking activity, pressures, and ecosystem towards adaptive assessment of the thermodome, which was supervised by Joël and Denis in Brest at the Social Science Institute Amur, and it was integrated in the Zagadome project, which investigates or explores hybrid ocean governance in the high seas. So what are the high seas or ocean areas beyond national jurisdiction? It's everything that we can see here in dark blue, and it's everything that is outside of the exclusive economic zones that usually expand up to 200 nautical miles from the coastline. While it covers over 46% of the ocean's surface, it is still underrepresented in conservation, and that's where the BBNJ treatment comes into play. BBNJ stands for Biodiversity Beyond National Jurisdiction, and it's over 100 countries, member states, that agreed on it. It tries to foster conservation in areas beyond national jurisdiction and to foster the sustainable use in these areas. In part 3 of it, measures like area-based management tools, ABMTs, inclusive MPAs, marine protected areas, are discussed. And now we will take a look at our case, the thermal dome and the eastern tropical Pacific that you can see here. So the thermal dome that you can see here in this area, in this like light blue, is a permanent offshore upwelling. That means cold, nutrient-rich water comes up to the surface and produces a rather productive area. It is defined by the 30-meter thermocline. That means that the thermocline is relatively shallow and the water comes up from the deep. Because these winds that we can see here in orange and the major ocean currents in yellow vary over the year, and in between years it is intra and interannual spatial variable. That means it moves all the time, it's a dynamic feature. We will take a look at this now in a little animation. The dark or the blue violet places in this graphic will be the shallow thermocline. So in the beginning of the year, it is closer to the shore and moves over the year offshore and has its highest band around September and October. So as you can see, it moves around in this area, and you can see here in the black lines the EEZ, so the exclusive economic zones of Costa Rica, Nicaragua, El Salvador, Guatemala, and Mexico. So it moves between different national jurisdictions, and also goes out of these national jurisdictions beyond. Meaning it is cross-boundary and it's rather hard to manage. That's why we need to take an ecosystem approach to it. And as we already talked about, it creates a high productivity and upwelling, and it was recognized as a very important ecological area. That's why it's a potential candidate for a marine protected area under the new BBNJ treaty, which needs a science-based rationale to support this proposal, which brings us to the science policy interface and an interesting framework. It's a framework to kind of assess a social ecological system. It's called the DAPSIA framework. We can start here with the drivers, for example, societal need for food that requires activities, which could be fishing in this case, for example, and these activities will cause pressures on the ecosystem, which can be species extraction or bycatch. Then, these pressures will have an impact on the ecosystem and create a state change of the ecosystem, which then impacts the human welfare, so reduces, for example, the ecosystem services. In the end, it's rather action-oriented and goes to the responses that try to tackle the driver activities and try to regulate those, or tries to mitigate the pressures. You can see it's quite a holistic framework, and it's really hard to apply. That's why I will only look at these 3 parts, and I will link activity, pressures, and state change to have like a first assessment of this social ecological system. So the objective of this work is to contribute to a DAPSIA framed assessment of the thermal dome by applying the integrated ecosystem assessment method. To, in the end, contribute to the synthesis of knowledge, to reveal knowledge gaps, and to prioritize threats. So, scoping an integrated ecosystem assessment is a semi-quantitative method. That means it includes quantitative and qualitative data, and it is especially good in areas where there's very low data and knowledge about it. It's a formal process that can be reproduced in other ecosystems, and it will provide a holistic data synthesis and analysis. In the end, it still can illustrate a concise view of interactions and threats in the social ecological system. Now we will take a closer look at the process of this method, and we will start here with the literature review. So we were provided in Brest by our partner Maviva that gave us like a gathering of the existing information of the thermal dome, and that was like the starting point. Then our project group, which was a transdisciplinary group out of people from France, Costa Rica, and Colombia, defined 8 different activities like fishing, shipping, tourism, for example, 9 pressures like targeted species extraction, anthropogenic noise, and 12 ecosystem state components, epipelagic fish, dolphins, whales, etc. In the next step, we looked which activity is causing which pressure. So that was the first step, and then the second step is which pressure is causing which, or is impacting which ecosystem state component. Out of over 900 possible links that were created, 321 links were identified in this ecosystem. And these 321 links I had then to score according to the spatial extent, to the frequency of occurrence, and to the degree of impact. Also, we assigned a confidence level that tells us how good is this link informed. I will give you maybe a better idea if I'm showing you parts of my metrics that I worked through. We will take a look at the one that I marked in the middle, and we have the activity shipping, we have the pressure incidental physical harm to whales. In the spatial extent is widespread patchy, that means it is widespread, but not everywhere in the same degree. And then the frequency is common, and the degree of impact is acute, as collisions with whales will cause significant injury and death in whales. And also we assigned a confidence index of 3.5, which is like average, because 5 is the highest and 1 is the lowest information. And here we have the sources that informed our decision. In the next step, we are here. We translated those categorical values into numerical values. And out of these numerical values, we multiplied it, and we had an impact risk that we had then for each link to assess. We again take a look at our matrix, and again at the link that we looked before on the right-hand side. We now see the numerical values, the impact risk score, and then the relative contribution of this one link. And this one, for example, has over 7%. That means it is rather a high risk in this ecosystem, so this pressure and this activity is rather dominating in this ecosystem. Then I took all these impact risks and analyzed them in an integrated ecosystem assessment report, which still has to be verified by a bigger and more broad stakeholder and expert workshop, which is hopefully happening in October or November, and they will verify and validate the work that we did. So, coming to our results, we have this beautiful Sankey diagram that shows the flows between, on the left-hand side, the activities, in the middle we have the pressures, and on the right-hand side we have the ecosystem state components. We can see, when we look at the pressures, the activities, that fishing is highly dominating the ecosystem by mostly causing bycatch and targeted species extraction. If we look at the pressures, we see that 5 pressures are dominating the ecosystem that are more or less in a similar impact risk. If we look at the highest rated one, that is just a little bit more than the other ones, it's incidental physical harm to species. And we can go back and see, okay, it's mostly caused by fishing. And what is the component that is mostly impacted? It's whales. So this we can like go along this flow diagram and see how the impact in this ecosystem, and how the flow of the impact is distributed. What I also did is a so-called gap analysis. I took a look at two of the activities, shipping and fishing, just for example now, and we looked at the number of links, meaning how many links or how many linkages were caused by shipping and fishing. And here we have our confidence level. So the upper two levels are based on expert consultation, and then we have in the middle, in the yellow, greenish one, global literature informed our link, and then going to the blue ones is regional literature, so literature on the ecosystem or on the region, and then we have the best, that is regional data and monitoring. And we can see shipping, there are a lot of data gaps, and mostly it is informed by expertise or global literature that is not yet that evident. And we have little links that are rather evident in science. Then we have fishing that show two links that are based on monitoring, but still the monitoring is not sufficient enough. That's why it's not in the best category. We can see that we have a better informed linkages in the fishing as there is more global literature on the different pressures. We will also take a short look at the pressures. Same thing, the pressures that were linked to these different links that we assessed. And we have anthropogenic noise, for example, and we see a lot of red. So there is still a lot of knowledge missing, and there is more studies needed. Same we see for incidental physical harm to species, that it's still rather badly informed. For example, target species extraction and bycatch is a bit better informed, but and also a bit more balanced. But we see still that also there is some knowledge lacking. What was proposed by ISIS is a simplification of what you've seen before of the flow diagram by only taking into account the top 1% linkages. That means every link that is contributing more than 1% to the total impact risk. And then we can see we can easier identify the key areas of interest, so we can prioritize, and we can especially, it's more accessible to people. Also, over 72% of all impacts are still represented. Coming back to the BBNJ agreement and seeing how was this work contributing to it, we look at Article 19, where the proposals are described. So how should the proposals be informed? And the proposals should be informed on the best available scientific knowledge or knowledge. It needs to take into account a precautionary approach and an ecosystem approach. We took into account an ecosystem approach, and the precautionary approach we also took into account by assessing each of the links, no matter how badly they were informed, and they could still have a big impact risk in this ecosystem. Also, key elements needed to be integrated. Human activities, which we can see here, the fishing effort, or the shipping lanes that cross the dome, the possible impacts, which were pressures in my case, because here, for example, the bycatch of sharks by the fisheries, or the blue whale sightings that overlap with the shipping lanes. And in the end, the state of the ecosystem, but it's rather hard to assess the state of an ecosystem, and that's why we took this rather holistic approach to look at the activity, the pressures, and then we can see the state change to describe, for example, several sharks are dying because of bycatch, or whales are dying because of collisions. To conclude, the aims to synthesize knowledge, to reveal data gaps, and to prioritize threats can be considered as fulfilled, as fishing and shipping are by far the most impactful activities that need prioritization in monitoring and management. 5 major pressures were shown in this social ecosystem, and especially anthropogenic noise and incidental physical harm were rather badly informed and need really more research. And these 4 ecosystem state components were under greatest risk. The limitations of this work, of course, we have some poor data areas and some insufficient consultants. We have, maybe you've realized, climate change is not included. This is because it kind of overlays everything and is integrated in further steps of an integrated ecosystem assessment, as otherwise it would be impossible to prioritize the activities and pressures. So, future of this work is a base for a validation workshop, which will also foster discussion and will validate the data and bring in the last expert consultations that were missing. Then we need to consider climate smart marine protected areas or marine planning, as well as projections for the ecosystem. And we can also use this as a base to do more focused assessments like this one, for example, only for shipping. Then we look at the different shipping activities, which countries were contributing, which type of ships were contributing, and we can do that by an expert group only focusing on shipping. And this can then really engage the policymakers and co-develop management measures, for example, for a proposal to the International Maritime Organization to regulate fishing activities in these areas. Thank you very much for listening, and thanks to all these people on the slide. Thank you for listening. These are the references, you can see them.
Thank you very much, Nikki, for this very interesting and well-done presentation. We're about 16 minutes, 30 seconds, so a bit over time, but it was interesting nonetheless. Thank you very much. Who wants to start? Estelle? Right, thank you.
Well, very interesting work we have to have on my for example, human needs a lot of data, right? So, what would be the hardest part of this work for you? I mean, when is the data, is the computer collect the data?
I would still say the collaboration, because it's just hard to manage a working group that is in different parts of the world with different time zones, and like the processing in the beginning, it was really good for defining the different categories, the activities and pressures. We worked a lot together. But for example, in Costa Rica, like 5 o'clock here in the afternoon is 9 o'clock in the morning in Costa Rica. So that was like the time of our meetings. And so we took like a different approach. I kind of assessed all of the links and sent them to to review and give comments. So we kind of changed the different procedures, how they are usual, but it also worked. But I would say this is also because I'm not yet done with my manuscript. Like collaboration just takes a lot of time. For example, this gathered information that was the basis, like for the literature review, was supposed to be there in January, but it came in like beginning of May. So there was a lot of like challenges with this, which I guess is very usual in science. And just if you have this frame of 6 months, it's a rather big challenge. Yeah.
Would you like to continue in this field, or?
Yeah, exactly. The idea right now is actually to organize the stakeholder workshop, because I'm also the person that is into this methodology and could lead this workshop. And we all met in Nice, in Nizza, 2 weeks ago, and we discussed this, and they want to go on with it. It's still not sure how like the financing is working, but the idea is to continue and at least to finish this first part of the integrated ecosystem assessment, as it's just like the beginning for more work to do and for also very different analysis. I don't know if the people meet in the workshop, for example. This also fosters collaboration between different disciplines, and that can like launch new projects on the thermal dome, which is like the goal of all the work. Yeah. Sure.
Well, congratulations for this work. I really like how it was framed and presented, maps were high quality, it was clear what we need, and I really like it. Congratulations for that.
Thank you.
And I have a question about the basis of this stuff. Like, why is there a thermal dome in that area? What are the oceanographic conditions that drive this dome? Because Do you wonder if climate change has an impact? To address that, we need to know the surrounding conditions that drive that on.
Absolutely. I mean, climate change does have an impact on it, and it's an extremely interesting area because it is highly impacted by the enso-phases, so by El Nino and El Nina. And for example, during El Nino years, the thermal dome is way, way smaller, meaning that if the surface temperature will increase over the years, it is likely that the dome is changing and is reducing. That's like, this area is super interesting to also do these kind of projections and experiments because we already have the data because El Nino has a high impact on it. I didn't include it in this presentation because it's kind of out of scope and it's like we need way more time. But the North Equatorial countercurrent is slowing down if El Nino is if we have El Nino conditions, and also the jet winds will lower down. So yeah, and so the jet winds and the countercurrent are driving this phenomena, and that's why it's like in El Nino years it's less extended.
And it's not seasonal, but it's all the year round.
It is seasonal, like you've seen in this animation, but it is still there somewhere in the region. Although if you take one point, it's not all, like over the years it won't be there. So like the persistent, the permanent is like it's somewhere, but it's not at the same spot. And also this is an average of 30 years that you're seeing. So there's a lot of variation, which makes it so hard to manage the system. But this like geographical area of collaboration gives the first like idea. I mean, it's based on oceanographic data, and you can still find like measures that are seasonal and that kind of move with the dynamics of the dome. But it's like far in the future. This is like really a base for more work on this region.
I guess that it could be not 44, but maybe later on another step. It could be more specific in terms of the space, the spatial distribution, because you have the distribution of the dome monthly, and then also you have the distribution of impacts, because we were showing there marine traffic, and probably you can get the position of the fishing boats. No, you can be more explicitly space.
Absolutely, you can, but the thing is right now, for example, the abundance data of different species is very, very like badly informed. And that's why we took this approach, and why we took the approach from the activities to the pressures to the state change, instead of looking at the state and trying to assess what is there and like how we can manage it. We really need to like take the step from the activities to have more information. But you're absolutely right. The next step would be to go into spatial planning. But this is more like also to prioritize what actually needs to be managed, because, for example, those graphs do look very different in different ecosystems. So it's not usual that fishing and shipping is dominating it. There's sometimes tourism that is totally dominating an area, especially in coastal areas. There's a lot of land-based activities like that is highly dominating the area. And even in the high seas or in areas beyond national jurisdiction, where you have these major ocean gyres, there's all the plastic pollution that is dominating the ecosystem. So those look very different, and it's like a first step to go on with the work that is in the future. Yeah.
Congratulations.
Sure. Thanks.
Congratulations.
Thank you.
Everything was really clear what is saying, so congratulations. Just it's more curiosity than a question. I would like to hear your opinion. I mean, you did this big work with Jeff to understand what's happening in the past. And to in the future create an MBA. So, what is your opinion? You believe that an MBA there could be created in the short term, but in the very short term, or because fishing and shipping is the real impact there, so maybe there is some problem with
Absolutely, like this is a huge problem. Well, the thing is, it already was shown in the last couple of weeks because like it's a rather geopolitical question and like a political question because Costa Rica said that they want to drive this proposal, but they want to do it all alone. So, and they don't want to include the different Central American countries, and under the BBNJ agreement, you would also need to include the different actors in the areas beyond national jurisdiction. For example, the fishing fleets here in the areas beyond national jurisdiction are Chinese or Taiwanese or Spanish. So it's like you cannot take them out of the process. And in the end, Costa Rica is kind of trying to foster this process, to have also maybe control over the area to manage it. But it's not the idea of the BBNJ agreement. I mean, it's in the text that you should collaborate and then that you need to collaborate with different states and different stakeholders. And that's like why we take like a side role right now, because they're trying to foster the process by their own. And I think the NGO that we work with is not in the best relation, but they were driving this process. Like a few months ago, the Costa Rican government wasn't even accepting that there is a high productive area. And now with like organizing with the others, the United Nations Oceans Conference, they had like this thing as their like little baby to put in front and to say we're doing something for conservation. But it's not like the process that like how it should be, and that's also the project that tries to explore what could be the implementation of the BBNJ agreement in terms of the spatial planning and the marine protected areas. So it is a very complicated and complex process. But we're trying to take like a more structured and formal way to go to this, to support a proposal instead of going like, oh, this could be a good opportunity for us to control or to manage this area. Well, we will see what will happen, but our goal is really to organize this workshop to bring together different people that are doing research or that are doing activities in this area, and to try to gather the best available knowledge, which should support a proposal and not just the political will of like maybe other reasons. Yeah. Sure.
Thank you very much. I had a question, but we're going to be short on time, so I'll ask you to coffee.
Okay.
Thank you very much, and congratulations.
Thanks.
My next presentation is from the Sorry, that's one of the Sorry. We still see you, Mr. Stanley.
Okay.
We still see you. We need to control the computer.
Yeah.
Whenever you're ready.
Good morning, everyone. I'm Gwendolyn, and I will present my master thesis work focusing on the green extraction of anti-cancer and anti-inflammatory compounds from mycology, supervised by Dr. Chiara Lontano at the Stazione Zoologica Entendorn in Napoli, in Italy. Have you ever thought that something invisible to the naked eye could actually help treating or preventing cancer? Well, that's actually the case of microalgae that are microscopic photosynthetic eukaryote that thrives in marine, brackish, and freshwater environments. And they are known to produce a wide variety of compounds from their first and second metabolism, known to have properties such as antioxidant, anti-age, anti-inflammatory, and anti-cancer. Microalgae are an excellent source and promising source for anti-cancer prevention, which is the leading cause of death worldwide. This study focuses on two marine plumate diatoms, Cyclithotheca closterium and Dithyramb brathwaite. The strain in the Sassonia zoological entomology for Cyclithotheca closterium was named AP2, and for Dithyramb brathwaite it was AP21. Cyclithotheca closterium is known for its anti-cancer properties and was tested on various cancer cell lines, and also for its anti-inflammatory properties, tested usually on monocytic leukemia cell lines, so called THP1. While for Dithyramb brathwaite, at date, it has not been tested for its biological activities. To extract those bioactive compounds, conventional methods such as Folch, lion dryer, and Soxlet remain widely used, and they require large quantities of organic solvent that are often harmful for the environment and leading to the generation of hazardous residues. And that's why new extraction techniques are starting to emerge, and they are referred as green, such as the supercritical fluid extraction or the microwave assisted extraction. And they require little to no toxic organic solvent, and they offer several advantages, such as high extraction selectivity, short extraction time, and improve the extract quality and the yield. The use of conventional extraction method to obtain compounds with anti-cancer activities and anti-inflammatory activities contradicts the fight actually against cancer, preventing or treating such an aggressive disease as cancer by using organic solvents that are harmful for the environment and for the human health highlights the need to prioritize more cleaner and sustainable extraction methods. And that's why the aims and the objectives of this thesis was to evaluate whether the conventional extraction method and the green extraction method exhibited different levels in the anti-cancer and anti-inflammatory activities, and was tested on various cell lines, and also to identify potential anti-cancer and anti-inflammatory compounds through characterization and literature-based analysis. To evaluate the anti-cancer and anti-inflammatory effects of my extract, I first needed to cultivate my algae. Thankfully, because of time restriction, the Sassonia zoological kingdom already had pellets available from Dithyramb brathwaite, so I only needed to cultivate Cyclithotheca closterium, and it was cultivated in 10 liter polycarbonate over 6 days at fixed parameters, and was harvested by centrifugation. The biomass of Cyclithotheca closterium and Dithyramb brathwaite underwent pre-treatment to disturb the cell wall and to release the bioactive compounds. It underwent the treatment were sonication and freeze drying, and it was stored at -80 until extraction. To distinguish between specific bioactivity and general toxicity, I also used normal human cell line, so immortalized keratinocytes and human lung fibroblasts, MRC5. For the anti-cancer screening, the cells were adherent cells, so human melanoma, A2058, and hepatocellular liver carcinoma, AP2. And for the anti-inflammatory screening, it was focused on with the THP1 cell, as I said before, the monocytic leukemia. Regarding the extraction, so I tested the conventional extraction was maceration with methanol, which is an organic solvent, and it was added at a ratio of 5 ml per gram of pretreated biomass, and left to macerate for 1 hour at room temperature. For the green extraction, I tested 2 still maceration, but with ethanol, which is a greener solvent, and can even be biosolvent when it's from the fermentation of sugar. It was added at a ratio of 10 ml per gram of treated biomass and left to macerate for 24 hours at room temperature. And the resulting supernatant, they were concentrated by rotary evaporation. The next green extraction technique that I used was supercritical fluid extraction using supercritical carbon dioxide. However, carbon dioxide is known to be a non-polar solvent, therefore it extracts better non-polar compounds to moderately polar compounds. And that's why I decided to add ethanol as a co-solvent that allows the recovery of more polar compounds. The supercritical fluid extraction was performed by Serena Lima at the University of Palermo because it's a really expensive technique, and our lab was not equipped with the machine. So I sent her 5 grams of dried biomass of each microalgae, and she proceeded with the extraction for 90 minutes at 200 bars and 60 degrees. Now that my extracts were ready, I was able to finally test the anti-inflammatory and anti-cancer effects of my extract. For the anti-inflammatory, I first seeded my THP1 in 96 well plates and differentiated them into macrophages with PMA. Then I treated my cell with my extract at different concentration, 1, 10, and 100 micrograms per ml, and was left for 24 hours. And then I induced an inflammation response with LPS and read the TNF-A inhibition thanks to the test. TNF-A inhibition is TNF-A. It's a signaling protein that is induced when there is an inflammation response. Regarding the anti-cancer assay, the same protocol was performed. So I stayed my cell in 96 well plates and treated with my extract at the same concentration, 1, 10, and 100 micrograms per ml, but at different exposure time this time. So 24, 48, and 72 hours. And the cell viability was measured thanks to NTT-assay. Regarding the results for the cytotoxicity activity, normal human cell lines, so on immortalized keratinocyte and non-fibroblasts, the result exhibited a species, extraction method, and dose-dependent response. After 72 hours of exposure, the ACA cell treated at high concentration exhibited low cell viability, so cytotoxicity. The cell viability was under 30% for all of them, except for Synprotecyclosporidium extracted with ethanol. At low concentration, for the supercritical extract of Synprotecyclosporidium and Dithylamb brightwelli, it seems to have promoted the growth of the cells. While for the non-fibroblasts, the cell viability was in general better, even if cytotoxicity effect was found for the supercritical extract at high concentration of Synprotecyclosporidium and Dithylamb brightwelli. And this result suggested specific bioactivity on normal human cell line. Regarding the anti-proliferative effect on cancer cell lines, so on human melanoma and hepatocellular carcinoma, the results exhibited an exposure time, extraction method, and a dose-dependent response. Indeed, after 72 hours for the human melanoma cell line, the lowest cell viability was reached at high concentration for the supercritical extract of Synprotecyclosporidium and Dithylamb brightwelli, knowing to be 26% and 19% for Dithylamb brightwelli. And for the human non-fibroblasts, the lowest cell viability was still reached for the supercritical extract at high concentration, but it was reached after 48 hours of exposure. The cell viability for the two extracts was approximately 25%. And regarding the anti-inflammatory assay, several extracts expressed an inhibition of TNF-A. However, non-significant results were found. But we can still see that at high concentration, cylindrodesa clostridium extracted with ethanol and supercritical free destruction exhibited a good inhibition of TNFA. And the same thing happened for diethyl botryoline extracted with supercritical free destruction. However, we can clearly see that we have negative values when our macroalgae were extracted with methanol at high concentration, which means that there was a stimulation of the TNFA production, therefore an inflammation. To potentially understand what are the compounds that have anti-cancer and anti-inflammatory properties, I decided to characterize my extracts, and I found that the characterization of my extract was species and extraction method dependent. Indeed, the pigments that were targeted, indeed the pigments, cylindrodesa clostridium, yielded in general higher quantities of pigment than diethyl botryoline among all the extraction techniques. However, for diethyl botryoline, it was the opposite for the fatty acids. It yielded higher fatty acids, and cylindrodesa clostridium yielded lower quantity of fatty acids. The method of extraction also influenced the recovery of bioactive compounds. Supercritical free extraction coupled with ethanol usually was able to recover all the pigments for both microalgae and was able also to recover all the fatty acids for both microalgae, while for the other extraction method, it was not the case. So supercritical free extraction was found to be the best techniques to obtain compounds with anti-cancer and anti-inflammatory activities, as we saw before in the results, but it was also found to be the most sustainable techniques. Indeed, I assessed the environmental sustainability of the supercritical free extraction, the ethanol extraction, and the methanol extraction of diethyl botryoline and cylindrodesa clostridium. So here we have the score of the supercritical free extraction of cylindrodesa clostridium and diethyl botryoline, and it has been found to be the highest score, and it's a score ranging from 0 to 1, and it usually takes into account, for example, the waste generation, the biomass loaded, the type of solvent used, and the energy consumption. The main bioactive compounds targeted among both microalgae were theophobin A and fucoxanthin, and they are known to have anti-cancer and anti-inflammatory properties. While for the fatty acids, the most recovered one were EPA, magnetic acid, and palmitoleic acids. In the literature, EPA, which are polyunsaturated fatty acids, are most studied for their anti-inflammatory, antioxidant, and anti-cancer activities. So we can possibly think that those compounds are responsible of the biological activities, but the fractionation of the extract could be a further direction to actually know if it is the case. In conclusion, this study explored for the first time the anti-cancer and the anti-inflammatory potential of cylindrodesa clostridium and diethyl botryoline using environmental friendly extraction techniques. Among the tested techniques, supercritical free extraction demonstrated the highest selectivity for anti-cancer and anti-inflammatory compounds and was found to be the most sustainable techniques. Indeed, supercritical free extraction exhibited the strongest anti-proliferative effect on human cancer cell line, but showed cytotoxicity effect on normal human cell line, specific cytotoxicity effect on normal human cell line. And finally, this study reveals the potential of diethyl botryoline to be, that is the nursery species, to be a potential source of bioactive compounds. And this study also supports the transition towards greener extraction methods. The future research that we should focus on is the fractionation of the extract to know which compounds is actually responsible of the anti-cancer and anti-inflammatory activities, to do metabolic profiling, also optimizing the upstream conditioning and the biomass stabilization that is going to actually influence a lot the production of bioactive compounds, and also to optimize the parameter of the extraction, and to know in-depth mechanisms for potential therapeutical application for CBD, cyclosterone, and titanium pathway. Thank you for the
Thank you very much for the meeting. We only have a minute over time, but good flow with better in the presentation. So thank you very much. So I'm going to go over to the members. You want to start? Right, thank you.
Yeah, congratulations for this presentation. I really like how it was presented, and all the results are very nice. It's such a big open topic, by the way. Although it goes a little bit beyond my expertise. So I guess that's the question because good question. And we still have not addressed the bioactive molecule, no? responsible of anti-cancer activity.
Sorry, can you repeat again?
You haven't isolated yet
No.
the bioactive.
That's like, yeah, that should be future
So
direction.
can you describe how would you go from here to achieve, from your results here to achieve that goal?
So to achieve that goal, yeah, because I know that the strongest anti-proliferative effect were found to be with the supercritical extract, I will take my supercritical extract at 100 microgram per ml, and I will fractionate, and then I will see which compounds it actually, and then test again on cancer cell, and test which compound has the strongest anti-proliferative effect, and know which compound is responsible for.
How many compounds, different compounds can be there to test?
In one fraction,
Yeah.
usually it's usually it's one, yeah. So it depends which solvent you use, and then you also need to quite know before which compound you want to target before fractionation. So it depends on literature-based research analysis.
Thank you.
Nico? Steph?
Again, congratulations for our work. It's amazing and a lot of behind that master thesis because you have used very different techniques. I mean, culture, chemistry, and cell culture. So my question is that the work of this work has to be done because it's different techniques and it's amazing to learn.
So the only thing that I haven't done is the supercritical treat extraction itself.
And the extraction technique that
No, the supercritical treat extraction. So the green extraction of the compounds was done by in Palermo, and also the characterization of the compounds. It was also done by Seven in the. It was all that I did all the rest.
what's the expense that you fix
I used ACAT, MRC5 for the non-malignant cell line, and A28, A2 for the anti-cancer screening, and THP1 for the anti-inflammatory.
Thank you, Steph.
Again, just a curiosity, congratulations for your presentation, and it's a very valid mind. But I just want to know if this extraction method, which is quite amazing, could be maybe used at a large scale, or you think it's something for now?
Yeah, that's why this thesis was submitted privately. We don't want to publish the results yet because we want actually to publish the thesis because it's the first time that we tested Nombatwili. It has never been tested. Well, it has been tested, but for the methanol extraction, and no anti-cancer effect was found. However, here with the supercritical treat extraction, we found really good, strong anti-proliferative effect. So we really want to publish and potentially how do you call it? Label? No. label this, but that's not my, that would be my idea.
Alright, I wanted this is completely out of my expertise, but what I recommend on the amount and the quality of the work you presented. I will ask you about a question of my expertise that you skipped over one of the first graphs you said you showed was the bar, the two bar, like multiple bar plus. It mentions in the legend like two-way, and this one two-way, and so what are the factors here?
So I grouped two-factor.
Yeah.
I looked the extraction method and the string.
Okay.
And it was compared to the control, so just something out.
So it's a one-way.
Yeah, but because I wrote them, my
No,
supervisor
you
said.
should say it's a one-way.
Okay.
If you have to have like switch, it's just presenting it if it's
Yeah.
one way, and you should have if you're having two-way, then you could be interesting for maybe to test the interaction, because that means the two factors will have the same effect, and you have that. Because I was just like, I was thinking,
Yeah,
that's
okay.
a one-way result of that,
Yeah,
and then
okay.
it just popped out there like that. That's great. Okay, so that answers my questions. Are there questions from the audience? We have some experts in biotechnology. They could be asking some questions. Don't be shy. No, you don't want to? Come on. That's very good.
Yeah.
Anybody else? No?
Yes, yeah, also curiosity, like from those experiments to application into like clinical experiments, how long is like the time frame? Is it like 20 years or is it hard to say?
I don't really know, but I guess that's really, really long. And also we need to take into account that, for example, for the supercritical feed extract, I needed 5 grams of dry biomass of microorganism, which is actually a lot large quantities. And I only cultivated cylindrotesaccosine, not dictylium between, and it lasts me almost 3 months. So it's parameters that needs to be optimized to reach like shorter process like time in general to go to clinical research trial. But at the time right now, we need more research also on normal human cell line because it is cytotoxic for normal human cell line. And you don't want to put something in your body that will also kill your normal human cell line.
Thank you. And I just thought of the
And you said that you already used ethanol for the extraction. But like you know the kind of the same lab, and we used ethanol and we also used water for the extraction. So did you use water together or did you use any other solvents for the extraction?
So conventional extraction was methanol.
Okay.
Green extraction was ethanol and carbon dioxide and ethanol. But I also know that alternative solvents such as water is also promising to extract, but it's also a polar solvent, so it's usually extract more polar compounds than non-polar compounds.
I have a question. The last slide, in your conclude slide, you showed some graphs. You said something about them, but you didn't explain how to read those things.
This?
No, again.
This?
Yeah, yes. Can you explain those things?
Yeah.
I was intrigued.
So this is a score that, so it's a free software where you can basically enter all of your parameters and it will give you a score ranging from 0 to 1 and you will know if your extraction technique is actually sustainable or not. For example, the first one is how much biomass you loaded. So it's quite a lot each time. And then I don't remember which one, but then it's the biomass, the waste generation. So it takes into account the pellets biomass, but also how much solvent then I discarded after. So this was the agri score for cylindrotesaccosine, supercritical feed extraction, ethanol extraction, and methanol
I
extraction.
remember, but I got the general score, but I didn't know how it was.
So yeah,
Okay,
supercritical
right.
feed extraction is really good, but I think we could reach an even better score if we actually optimize the parameter of the extraction, supercritical extraction.
All right. Thank you very much for the evaluation. We will be taking a break, and for those following online, presentations will resume
those questions.
review. If you want to move, to show something on the board, you know that. So this is the limit of the command. Also, I don't need to show it. Also try to split. Yes. Right.
Here?
Yes, that's me.
Your last name?
Kempen.
Okay, just knowing if the pronunciation is correct.
Where are you?
I'm Hyde.
Open the door for invitation. Yes.
Ready?
I'm ready.
Okay, everybody, Dr. Heidi Kumar from HU will be sharing this session now. We are going to have this 3-minute Dr. Nicole Winter from University of Oviedo, Dr. Monida Correa from EPM, and Dr. Sandra from EPM as well. And then Olivier Kempen is going to present his thesis about fisheries conflict. So you have 15 minutes, and then we have 15 minutes for questions. So, okay,
Yeah. Is there like a way that you like announce that it's almost time?
Again?
Is there like a way to keep track of time?
I'll get up when you have 2 minutes left.
Yeah.
Okay?
Okay, we're ready. So, hello everybody, my name is Olivier Kempen and today I'm going to be presenting my master's dissertation. First of all, a big thank you for attending and giving me your full attention. So, the what and why questions. What is my research about? My research is about international fisheries law and geographical shifts that cause fishery conflicts over shared resources. The goal is to link such shifts with existing limitations of the existing legal framework and to propose future solutions to prevent such conflicts. It's relevant because in the last couple of decades the amount of conflicts has been increasing and climate change, namely the increasing sea temperatures, has been increasingly seen as a major factor contributing to those conflicts. I chose this topic because of my shared interest in marine law and marine sciences, which combined gave me a lot of knowledge about the challenges that fisheries are facing today. So this led to following a research question: how do climate-driven shifts in the geographic distribution of fish stocks expose limitations of the existing international fisheries law framework? And in what way do those limitations contribute to the risk of fishery conflicts? To tackle this, I used a layered approach where first of all I was going to examine the existing framework to look for limitations that were exposed by such shifts. Then I dove deeper into 2 case studies and analyzed them to gain deeper insights and to compare the findings with the first layer. And then lastly, I combined the lessons learned from the first 2 layers and combined them to look forward and to make propositions for future solutions in the Arctic Ocean. So first of all, the international legal framework. It's really a network that's surrounded around the United Nations Convention on the Law of the Sea. The most important features of this body are the distinction between the exclusive economic zone, where states have sovereign rights regarding fisheries, and the high seas, where there is a freedom to fish. There is a part devoted to dispute settlement procedures, which play an important part in fishery conflicts, but more about that later. Then you have the United Nations Fish Stock Agreement, where the main thing that you need to know is that this emphasizes the precautionary approach. This really means that in light of climate change and shifting stocks, states need to be cautious when implementing actions regarding fisheries, when scientific evidence is uncertain or inadequate. Then you have the United Nations General Assembly and the Food and Agriculture Organization. These are advisory bodies who provide guidance but they are non-binding. And then lastly, the regional fishery management organizations, which are seen as the primary bodies for international cooperation, who vary greatly, but all have the same goal, namely the sustainable harvest of fish stocks. Within this framework, each state has different duties and obligations. First of all, the duty to cooperate, which is needed to comply with the obligation on conservation of living resources. And then there is an important distinction that needs to be made between the obligation of conduct and obligation of result. An obligation of conduct is an obligation that is defined by making an honest endeavor, notwithstanding the outcome. So good examples are the precautionary approach, because it's not a pre-described result that needs to be achieved, and the duty to negotiate, which differs from the duty to reach an agreement, which is one of result. So these obligations of result, they are defined by having a pre-described outcome, where a good example is the determination of the allowable catch based on the best scientific evidence by a coastal state within its exclusive economic zone. So the limitations and the challenges to long-term cooperation lie first in the future uncertainty and economic incentives. Why? Because shifting stocks can change the cooperation environment in the future because different relevant states can become involved, and also the changed zonal attachment. But this will become clear in the case studies. But because these changes are difficult to predict exactly, this can lead to a future uncertainty and a harvest now mentality, where states will prioritize short-term gain over long-term sustainability. And then there are the limitations of the legal framework. The first one lies in the consensual nature of international law, namely the pacta tertis principle. This really means that it's not possible to require an unwilling state to join an agreement and to be bound by it, which can introduce free riders who can undermine and jeopardize long-term agreements regarding shared resources. Then there is also the interpretation of the obligation of conduct. Because there is no clear defined outcome, it's very difficult to assess whether a state has complied enough to this obligation or not. And also there can be a little ambiguity in the interpretation, where one state can claim it has done enough while another state claims it did not. Regarding the obligation of result, an important one is discretion regarding the determination of the allowable catch based on the best scientific evidence available. Nowhere in the framework climate change is explicitly stated to be included. So arguably you could say, okay, but it should be included in the best scientific evidence available. But because of discretion, which basically means that states have freedom to adopt measures as long as they comply with the result, this is kind of a gray area and difficult to check. And then you have the limitations in dispute settlement mechanisms under the sovereign rights that coastal states enjoy within their exclusive economic zone. This basically means that any obligations related to fisheries, when this happens in the exclusive economic zone of a coastal state, you cannot really do anything about it if the state doesn't comply with it, because it falls under these sovereign rights. And then there is the restrained scope of regional fishery management mandates. There's basically a mismatch between the authority of regional fishery management organizations, because they are ecologically and or geographically restrained, but the effects of these shifting stocks, they cross the borders, so there is a mismatch. Then to dive deeper into the case studies, first of all, the macro war. The macro war was restrained to the scope of the Northeast Atlantic Fishery Commission, so it's important to know a few of these provisions that are in place. First of all, the NIFC does not lead the quota allocation. The responsibility for this is placed at the states, who have to agree upon it in the form of coastal state agreements. When a coastal state agreement is reached, then the NIFC will adopt it. Another important provision is the objection procedure. This basically means that if the NIFC wants to imply or adopt recommendations or the coastal state agreement, states have the opportunity or the possibility to object to it and thereby not be bound by it. So the war itself, historically the mackerel fishery was done by Norway, the European Union and the Faroe Islands, but in the early 2000s the seas warmed, shifting the stocks northwards into the waters of Iceland and the Faroe Islands. In 2008 the war started when Iceland became one of the major harvesting states, and at that time they were given a little piece of the quota allocation. in the Coastal State Agreement, but when it was adopted by the NIFC, Iceland objected to it because they found they had a bigger claim to a bigger quota allocation. A year later, Faroe Islands saw these actions and decided to do the same, also setting unilateral quotas. This led to displeasure and sanctions of the European Union, and also closing ports to Icelandic and Faroese vessels. The war was temporarily resolved in 2014 when these sanctions were lifted, but revived when Greenland later joined in 2019, and together with Iceland again set unilateral quotas. A year later the Brexit happened, which also implicated some difficulties with access to UK fishing grounds. So as of today there is no final solution. Important is that between 2019 and 2024 the total summation of quotas exceeded the total allowable catch, which was advised by ICES, with an average of 130%, thereby imposing a challenge on the sustainability of the fishery. The next case study is the Pacific Salmon War, which happened on the west coast of North America, and was a war between USA and Canada. It involves Pacific salmon, which is an endonomous stock, and during their migration they cross boundaries of the states. Thereby it's inevitable that each state will intercept an amount of the other state's salmon. To manage these interceptions and the overall management, the 1985 Pacific Salmon Treaty was agreed upon, where the main thing was the equity principle. This basically means that each state should receive benefits equivalent to the production of salmon in their own waters. But this later was the source of conflict due to an ambiguity about the interpretation of it, because the equity principle was defined as an obligation of conduct. This ambiguity occurred due to environmental changes, which include closely spaced El Niño southern oscillations, increasing the temperature of seawater in the south, in front of Washington and Oregon, and shifting stocks towards the north in the Gulf of Alaska, which had more favorable conditions. Alaska then interpreted this equity principle as, okay, there are more fish in my waters, I can harvest more, which inevitably also led to more interceptions of Canadian salmon. On the other hand, Canada then interpreted this as, okay, you intercept more of my salmon, I can also intercept more of the USA salmon. But because stocks were already severely depleted in the south, this created a lot of tensions, eventually leading to a physical blockade of the Alaskan Ferry in 1997. But 2 years later, a revised PST marked the end of the war. So to conclude and to look back at the first layer, the Mackerel War was characterized by a change in zonal attachment and relevant states with the inclusion of Iceland and Greenland. And the objection procedure is really the embodiment of the Pacta Tertis principle, because it basically means that if a state is not willing to cooperate or be bound by an agreement, it can just object to it and become a free rider. And then lastly, there is the determination of unilateral quotas, which involve a lot of discretion, because it's difficult to check whether climate change was included. This could have led to lower quotas when climate change was actually included. And then the Pacific Salmon War, which was clearly about the ambiguity in the interpretation of the equity principle. So the future solutions of the Central Arctic Ocean. Why this ocean? Because it has a unique context. Currently there is no existing regional fishery management organization in place, and also the relevant states agreed to impose a ban on commercial fishing of 16 years until 2037. Therefore, there is a unique opportunity to build a regional fishery management organization from scratch. So to link the solutions to the specific limitations, first of all, the Pacta Tertis principle is really a fundamental principle in international law. Therefore, the best way to cope with this is to make states willing to cooperate by making cooperation more beneficial than non-cooperation. Important concepts are the new entrance strategy, because we saw in the Mackerel War that the inclusion of Iceland and Greenland led to conflict, and decision-making processes, because also in the Mackerel War we saw that the objection procedure is not really working because it's making it too easy to become a free rider. A proposed solution could be to allow objection, but only based on legitimate claims, which is already done by one other RFMO. Then regarding the discretion of climate change, this can be fairly easily solved by just explicitly including climate change in the best scientific evidence available. And regarding the interpretation of the obligation of conduct, Transparency is a very important thing to reduce the ambiguity. And also, a proposed solution could be a hybrid obligation, where instead of seeing the precautionary approach as just one of conduct, you can include sub-goals of result that can allow to keep track of whether a state has complied with the overall conduct or not. Then, regarding the restricted mandates, it's very important to improve cooperation with neighboring regional fishery management organizations, where the focus lies on dynamic agreements, where they can have overlapping mandates or a joint scientific committee, really increasing the responsiveness for future changes. And then lastly, regarding dispute settlement limitations, an Arctic Council could invoke binding measures or decisions, where the goal is not to restrict the state's sovereignty, but really show that if somebody does not comply, even within their own waters, there will be consequences which can act as a motivation to cooperate. So to end the discussion and the conclusion, first of all, several limitations that were identified are not restricted to fisheries law, like the pacta terris principle and obligation of common result. They are also applicable in other branches of international law and also impose implications there. It's also difficult to generalize the exact cause of conflict because there are a lot of other factors playing, like politics or economics or cultural fisheries or historic fishing rights, etc. And to conclude, the geographical shifts expose various limitations that can lead over conflict over shared resources. But the main challenges are to reduce future uncertainty, improve transparency, and improve the willingness to cooperate. So lastly, I want to thank you in the audience, of course, my supervisor, my colleagues, students, the IMBRC board, and my family and friends who are watching. So thank you for listening.
Thank you for the presentation. I just maybe I didn't get it, but at the end, these solutions that you find were maybe based on an analysis of marine spatial planning or whatever, or this was just a big review and all the knowledge you have on this area and these cases.
So yeah, there is not much existing literature about specific solutions. How I did it, I first identified limitations and only looked at that because it's very broad. But I did it by looking at other regional fishery management organizations, and also as I found one paper that explained the ideal regional fishery management organization, and by combining existing things like the objection procedure, which is based on legitimate grounds, is already adopted in one of them. And I combined it with how an ideal fishery management organization would look like, and then I combined both to propose merely concepts, not very specific, but a concept that could work.
You use some criteria to identify the limitations and solutions, or define the criteria. I don't know, you collected from the literature or from the policy. And you identified some criteria to which to say, okay, I use this to identify the problems.
I'm not sure if I...
For example, when you make a review, you decide some criteria to which you select some papers and not others.
Yeah.
So you explain why you chose this. So the limitations were chosen based on what?
Did I identify the limitations?
Yeah.
Oh yeah. Yeah. So our work was, I first really read a lot, papers, the frameworks, the agreements, everything, and then there is an existing papers that talk also about things that are that can create sources of uncertainty, or it's a gray area in international law which can create fishery conflicts. So I had a list of concepts that can be identified as limitations, And then I read more about like case studies and yeah, relevant literature, and then I limited down the list to 5 things because there are more, but these are not really relevant to what I was looking for. And then I think the case studies really exemplified how this were really limitations in real life. So I think that's, yeah.
Yes. Thank you for your nice presentation. We talked about climate change in your presentation, but we defined some specific parameters. So climate change is a very huge word, so I want to focus on temperatures.
Yeah, I didn't really I really didn't mention it, but I really focused on increasing sea temperatures as a reason for shifting stocks, because I know acidification and oxygen levels also are a result of climate change, but case studies were really based on conflicts that were caused by shifts due to an increase of temperature of the sea. Like the Macro War was really scientifically proven that climate change was the reason, and then in the Pacific Salmon War, it was the southern El Nino oscillations that increased the sea temperature, thereby shifting northwards.
Your thesis is a question. So how do you compare shift in direct distribution? So do you find an answer to this question in your opinion?
Sorry?
So your thesis
Yeah.
is presented like a question.
Yeah.
At the end, which is your answer to this question?
So how do these shifts
Yes.
Yeah, can lead to fishery conflicts?
Yes.
The shift, I think it's because the frameworks were established a long time ago. I mean, the UNCLOS was in 1982, UNFSA was in 1995. At that time, there wasn't so much evidence or scientific research about such shifting stocks. So in my opinion, it's like the framework is not really adapted to it yet. So I think that's the main reason, that because of the outdated frameworks, these shifts are like too big of a change to tackle with the current framework. So there needs to be a catch-up process to really cope with and be responsive to future changes like this.
Okay, well, thank you very much for your presentation. It was a good presentation. You have completed and it could be followed perfectly. In my opinion, the presentation has to be a little bit more visual than your presentation. There are a few figures, too many tables, too much text in general. There is I think this one we need to put more figures down, more into the maps, or more symbols, also to be more visual. Not usually the one who is doing the presentation to the explanation.
Yeah, I think it's like also the nature of my study. I know, yeah.
But there are always ways to transform text into visual.
Yeah, you're right. Yeah.
And on the other hand, I also need a little bit of climate change in this study. We focus a lot on the legal aspects, and that we don't pass by very fast through the main climate issues that you mentioned. You just only mentioned that you didn't explain an Indian oscillation, for example. You didn't explain that the Indian oscillation is changing because of climate change and getting more abrupt and more unpredictable. So, likely in the future, there will be more fishing wars because of that. I miss the climate part of all. Did you, along the way, did you think in adding more climate stuff in the presentation? I don't know the answer, but I see in the presentation.
That I would include more of the climate change.
What's the climate aspect? Yes.
Yeah, the thing is, I didn't really dive deep into the climate change. I really saw it like as a leading factor, like rising sea temperatures can shift stocks forwards, and I took that as the reasoning, as the driver of conflicts that I was going to examine. So, yeah.
A couple of slides telling us what the need is and how it's changing.
Yeah,
That would be good.
Yeah, I had like an additional slide prepared. If there was a question of El Nino, then I would like explain a little bit. But I didn't think it was like really the focus of my thesis. But I understand because we're still learning science. I understand.
Sure. Okay. Congratulations.
Thank you.
Okay, we have time for questions from the audience. Is anyone here? Margarita? Okay, so I have a question. So if you could do this study, so your study was basically a test study, right? So if you could do this study in a more practical way, what would be your framework? How would you do that?
That's a good question. I'm not sure. I think in a more practical way, you get maybe like a set includes climate change more. Like really try to maybe figure out the case study yourself on how, at what rates do fish stock now exactly react on temperature changes, because there is a lot of studies about the rate of changes and things like that, but if you could do it yourself, maybe that add some additional value. I didn't really think of it, so I'm a little bit on the spot, but
Okay, what about the conflict part? How would you do that?
maybe like include interviews or like stakeholder meetings, focus group, and such kinds. But that's very difficult because these conflicts are really decided by like high-level persons with high positions. So as a master student, I don't really think that's possible. As a PhD, it would be more possible to like get interviews and like really get insights in how the negotiations are happening and things like that. Okay, thank you.
Yes. Sorry, presentation.
So we have Fortune presenting a little bit about invasive biomass being used for sustainable cosmetic production. So whenever you're ready.
Good afternoon, everyone. My name is Fortune Shiombola. Thank you for being here, and I will be presenting my thesis on bioprospecting and valorization of invasive biomass for sustainable cosmeceutical development, supervised by Dr. Luisa Custodio. The economic and ecological impacts of biological invasion, which we know refers to the spread, dispersion, proliferation, and establishment of non-native species outside of their natural habitats, is not new to the scientific community. They are often introduced via human-mediated pathways, and the impacts of invasive species are extensive. The marine ecosystem is highly vulnerable to these impacts due to the presence of high economic activities that take place at these intersections. Therefore, mitigation efforts towards uneradicable biological invasion must move from conventional to non-conventional, and this includes the valorization of invasive species biomass, turning them from waste materials into useful commodities, and this is implemented and serves as the overall aim of this study. The cosmetic industry has evolved from a purely aesthetic to a science-backed sector, driven by advancements in biotechnology, dermatology, and green chemistry, that has led to the rise of cosmeceuticals, which are cosmetic products with biologically active ingredients that offer therapeutic benefits like antioxidants, anti-hyperpigmentation, and anti-aging. In this work, we will we investigated 3 highly invasive species in Portugal, the Algarve, and now in other parts of the world, namely Acacia salina and Carpobotus edelich, which are halophytes or salt-tolerant plants, and Rubulopteryx ocamuri, which is a brown macroalga. These species produce diverse secondary metabolites with potent bioactivities, as we can see in this diagram. So, in this work, we extract and compare bioactive compounds using food-grade solvents. We evaluate the antioxidant and enzyme inhibitory activities of the extracts. We characterize the chemical composition of the extracts. We assess the interactive effects of the mixture of the extracts. We formulate prototypes right here using the most active extracts, and we demonstrate the potential for upscaling via a business plan. Now, over to the methods. To begin the extraction, the sample was collected and cleaned to remove impurities, and then oven dried to remove moisture while maintaining thermolabile bioactive constituents. Biomass of the individual species, so that's each of the species, and the biomass of the mixture of the 3 species were powdered, weighed, and extracted using water and ethanol coupled with ultrasound-assisted extraction. Then, the extracts were incubated and then centrifuged and filtered to remove residues. And then the water extracts were freeze-dried, and the ethanol extracts were concentrated via evaporation in the rotary evaporator. And then the extracts were resuspended in their respective solvents, water and ethanol, for further downstream processes. Then, extracts were characterized and quantified using LC-HRMS mass spectrometry, and the biocompounds were identified using feature-based and in silico tools. Then, the extracts were tested for bioactivities. The antioxidant activities of the extract was tested using DPH, ABTS, and FRAP assays. Second, the enzyme inhibition of the extract was tested on tyrosinase, elastase, and lipase enzymes. And then the ethanol extracts of the A. salina, R. ocamuri, and the mixture of the species was tested for cytotoxic effects via MTT assay on mammalian cells, namely hepatic carcinoma cells, which is tumoral, melanoma cells, which is tumoral and the main focus of this project, human neuroblastoma cells, which is tumoral, and human embryonic kidney cells, which is non-tumoral and also the focus of this work. The absorbance was determined spectrophotometrically, and statistical analysis was assessed. The cosmeceutical formulation for the lotion was developed using glycerin as a base, so that's what we have here. And then the gel was developed using carbopol 940 and HPMC as the base, which is this thick one here. To the results, the extraction method selected in this work was based on the solvent's efficacy in recovering a diverse range of bioactive compounds, also its grounding in green chemistry principles, and the potential application of the extracts as cosmeceutical agents. This aligned with the work of Saka and Nahar in 2012, encouraging the utilization of multiple solvent systems coupled with UAE to recover the broadest spectrum of bioactive compounds. After the phytochemical analysis, 82 and 55 compounds were putatively identified in the water and ethanol extracts respectively. Phenolic compounds emerged as the dominant class in A. salina, C. edelich, and the mixture of the extracts, and the lipids emerged as the dominant class in R. ocamuri extracts. Some of the potent compounds identified in A. salina, C. edelich, and the mixed extracts include catechin and quercetin, which have been documented to have strong antioxidant, photoprotective, anti-inflammatory, and skin brightening properties. Compounds like phospholipids and ceramides found in R. ocamuri extracts have been documented to have skin conditioning properties that aid in skin hydration and skin barrier integrity. So, the results of the antioxidant activity overall showed that ethanol extracts exhibited the highest antioxidant properties. A. salina ethanol extracts exhibited the highest reducing radical scavenging activity and reducing potential in ABTS and FAP assays. C. edelich ethanol extract exhibited the highest radical scavenging potential in DPH assay. And overall, R. ocamuri extracts in both water and ethanol exhibited the lowest antioxidant potential. So the high antioxidant property in ethanol may be linked to the presence of phenolic compounds that have been documented to have strong antioxidant properties, while the low antioxidant activity seen in arocamore extracts may be linked to the presence of lipidic compounds that are structurally less suited for radical scavenging activity and reducing potential. The interaction coefficient value of the mixed extracts showed synergistic interactions across all assays, which means that the mixed extracts performed better than the individual bioactivities. And however, in ethanol extracts, the IC value of the mixed extracts had synergistic interactions in both APTS and DPPH, but antagonistic interaction in FRAC, which means that the mixed extract performed less than expected based on individual bioactivities. In the enzyme inhibition assay, the ethanol extract exhibited A-salina ethanol extract exhibited the highest tyrosinase inhibition, and this can also be linked to the presence of phenolic compounds like rutin, myricetin, and camphorol derivatives that have been documented to be efficient tyrosinase inhibitors. And tyrosinase is an enzyme that plays a key role in melanin biosynthesis and has implications in hyperpigmentation. Our ocamore ethanol extract exhibited the highest elastase enzyme inhibition, and this may also be linked to the presence of lipidic compounds that have been documented to modulate elastase activity through hydrophobic interaction and steric hindrance at the enzyme's active site. The lipase assay unfortunately showed no quantifiable results, and this may be linked to the selectivity of the lipase enzyme and its substrates. The IC value, interaction coefficient of the mixed water extract, showed no interactions for the water extract. However, in the ethanol extract, the IC value only showed antagonistic interaction in the tyrosinase assay, but this result remained unclear and is open to further study. So, given their high antioxidant and enzyme inhibitory activity, the ethanol extracts of A-salina, arocamore, and their mixture was tested on tumoral and non-tumoral cells for cytotoxicity. In this work, I only highlight the two cells that are focused. So this is the human embryonic kidney cell, and this is the melanoma cell. And we can see highlighted in brown that A-salina extract exhibited a safe cytotoxic profile, which means that the extract can be used in cosmeceutical formulation. And we can see because there is high cell viability. Our ocamore, on the other hand, exhibited non-selective cytotoxicity, and this is due to the presence of lipids as well, which have been documented to act as cell irritants that induce cell death via intrinsic and extrinsic pathways like lipid peroxidation and mitochondrial dysfunction. The mixture of the two extracts significantly mitigated the non-selective cytotoxicity of our ocamore with high cell viability, and this can indicate a cytoprotective effect. The IC value, which shows antagonistic interaction, further highlights and confirms the buffering and cytoprotective effects that we see, and this cytoprotective effect can be linked to the interactions between the phenolic compounds in A-salina and the lipid compounds in arocamore. So we also assess the physicochemical characteristics, specifically viscosity, torque, and pH, to evaluate the formulation's suitability for use as topicals. Viscosity refers to the resistance of the formulation to flow, and torque refers to the force that is required to spin the spindle of the viscometer during viscosity measurement. As we can see highlighted in the deeper blue, mixed carbachol gel, which is this here, had high viscosity and torque values, and it indicates high shear resistance and thicker consistency, and it highlights the potential of this formulation to be an occlusive with longer retention time on skin and controlled release. As you can see in the HPMC gel right here, they had lower viscosity and torque values, which can indicate lower shear resistance and fluid-like consistency that highlights the potential of the gel to be used as fast-absorbing formulation where like quick and thin spreadability is prioritized. As expected from the lotion had the lowest viscosity and torque values, and this is typical for products designed to have rapid application, quick drying, and frequent reuse. The pH of topical formulations play a key role in influencing skin barrier integrity. Human skin maintains a slightly acidic pH ranging between 4.5 to 6, meaning that formulations must be within this range to prevent the risk of skin irritation and disruption of the skin barrier. As you can see in these results, the formulations containing the mixed extract exhibited a safe dermocosmetic pH range, meaning that they can be used in daily skincare or for daily cosmeceutical use. And the formulations containing the A. salina extract had elevated pH. While this doesn't necessarily mean that the formulation cannot be used, elevated pH have been documented to increase the risk of skin irritation and skin barrier disruption. But this can easily be fixed by incorporating pH buffering agents like lactic acid or citric acid in small amounts without impacting the texture or the bioactive stability of the formulation. For the sake of confidentiality, I can't speak too much on the business plan, but the business plan highlights Biodome as a sustainable biocosmetic brand that repurposes invasive marine algae into multifunctional skincare products. And the brand combines direct sales, business-to-business partnerships, and circular innovation to meet the rising demand for eco-conscious, ethical products while promoting marine conservation and profitability. In conclusion, the thesis demonstrated the cosmeceutical potential of 3 invasive species from the Algarve coast, and future directions include validating the interactions of the mixed extracts, in vivo assessments of the bioactivities, optimizing the formulation for all skin types, and responsibly upscaling production. I would like to thank these organizations for their support and my family and friends who are watching. Thank you, and thank you all for listening.
All three species are unique species.
Yes.
So, how much of these species we use to, for example,
obtain this? So, we measured. So, because we're not doing like a large scale production.
Okay.
We used a little amount for just enough for a vial, and it was about 10 grams for each species, and then we extracted and made the formulation.
And with the extraction, you have the waste, no? So,
Yes, we have waste.
The waste is basically almost all the amount that you use, and if so, if this has to be upscaled. No?
Yes.
What do you do with this waste?
Yes, so if this is going to be upscaled, several things that we can use the waste for. For example, the waste can be used as fertilizers. Mm-hmm. They can be used, for example, studies have shown that arocamerate, the marine algae, is used as compost for biocomposting. A. salina extracts, the A. salina, maybe I can just quickly go back to the picture. Yes, this is a nitrogen fixing legume. The waste can be used for fixing nitrogen into the soil because they are very good. They grow well in poor soil environments. Also, the waste can also be used for biopolymers in making specific materials, fibers, because the biomass at the end of the day, We can also use it to make other products. We can use it to make clay like masks, like facial masks, and also they can be used in nutraceuticals because people who take powders, they can be dried and used in cosmetics.
Which part you use the flower or the entire?
Yeah, so we use the entire plant because we didn't want to be too specific with the leaves or the flowers, so we just used everything together.
Thank you for your nice presentation. It's very, very interesting. What about the extraction? So do you find any difficult to extract the biocompounds from the biomass?
Yes, we had a bit of difficulty, particularly with the water extracts, because ethanol generally, it's highly soluble. But the water extract we had, that's why we had to centrifuge, because we couldn't just filter. We were losing a lot of biomass, so we had to centrifuge and multiple times to be able to get the most of the bioactive compounds that we wanted to extract. So that was the major difficulty that we had, mostly with just the water extract. The ethanol extract was fine, everything went smoothly, and the water extract, we just had to include centrifugation into the protocol. So, because normally they don't centrifuge. Yeah.
And how much biomass do you need to find some results?
Like results? So we originally used 10 grams of each, and then we did an even ratio for the mixture. But we just approximated that we'll probably lose half during the extraction process because, of course, by the time we centrifuge, we're losing some. By the time we dry, we're losing some. So at the end of the day, it's roughly about 5 grams that we need to make a simple vial.
And I was very impressed by the results of the mixture.
Yes.
So what do you think about this? So the synergistic effects of these three So it's something that you find with literature, something new?
Yeah, so it was very new for us because we searched literature and we couldn't really find a lot of information on interactive properties. We've seen literature where they discussed combining the extracts, but they didn't really say much on whether the interaction was synergistic or additive or antagonistic. But we decided to take the leap and hoping to see good results, and we found decent results. And for that research, we decided that the most plausible explanation for the interactions we're seeing, and our results also kind of explain, because if you look at the values for each, you find that this kind of does better than this. It's much lower than, does better than this. This is slightly higher. The same for this. It does higher than the C. edulage extract, and it's similar to this. Likewise here. So in places where we see antagonistic interaction, we see that, like in this case, the antagonistic interaction, it's much higher than these two extracts values, 78 and 178. So that also helped us to know that, okay, what we're doing, the results make sense. So we just know that the synergistic interaction means that probably the mechanistic pathways involved in the mixture of the two species were mutualistic. It was a mutualistic, one species wasn't trumping another, if that makes sense. Yeah, but in this case, it was easy to tell that one of the two species, or one of the three species that were mixed, was trumping the other in terms of activity.
Thank you. Thank you very much.
Okay, yeah, I really like your presentation.
Thank you.
Flowed very well, actually. Congratulations for that. Also, it's a very nice and useful presentation.
Thank you.
And especially at the beginning, because later on, for example, this figure here, you miss a chance to put a nice bar graph. So instead of this table that is more visually appealing and more much easier to explain.
Yes.
Well, we were now explaining this is higher than this one, so much better in that.
Graph.
than figure, than data, right? Do you think it is?
Yeah, so the initial thought was to use a graph,
Yeah.
but because it's going to be published, my supervisor advised to put it in tables.
Really?
Yeah.
I think it should be.
I mean, that was the first thing that I did, but yeah.
And also, I wanted to ask about the stats. Which stats did you use?
Oh, we use one-way ANOVA
One way ANOVA.
with and Kruskal-Wallis and the parametric and the non-parametric test. Yeah, after we of course check for homogeneity, if the data is normal, yeah.
Okay. I'm talking about the voltaics.
Yeah.
It's an amazing species
Yes.
here in Europe. So I guess your aim is actually to remove it.
So technically, we can't remove it because it's almost uneradicable. So these species are species that are uneradicable. So the only way we can, quote and unquote, remove is to just use them for something like better, because it's almost impossible to remove. To like completely remove, like, oh, no more Rhodopteryx, no.
But what do you think is going to happen in the future? This product gets commercial and it's a good product. Do you think that later a company will be tempted to harvest the algae?
Yes.
Yeah?
Yes, I think there is already like, I know in France, in Western Brittany, they are harvesting sustainably using like the Scooby-Doo method where they go and then they pick, but they have permits. So the only problem that could arise in the future is getting permits, because some of these species is not everywhere that they have been legalized to be sold to consumers, but some parts of Europe, France, I know of France because I've seen products that contain brown algae.
What's your personal opinion on harvesting?
For me, I just
the species.
yeah, I think it's good to harvest because the effects that they have, it compounds, it just keeps increasing over time. And the Okamure, the temperature is rising, so they love like the warm temperature, so it's just going to keep on, they're just going to keep on like growing. So harvesting, the only thing is we just have to harvest them sustainably in a way that doesn't destroy like the marine ecosystem, like the benthic ecosystem where they grow from. That's like the only way. But there are several methods that are being like implemented. I know of the Scooby-Doo method because that's the one that is widely used by industry standards, because you don't dredge the floor by, yeah.
But if eradication would have been possible,
it would be great.
Would you go for eradication?
Yeah, I would go for eradication. I would, yes, I would go for both. I would go for both, yeah. Well, I would go for eradication, but the company, in a way, it's a 50-50, the company will have to decide, am I choosing sustainability over money, or am I, choosing money? Because if they're choosing money, of course, they won't want yeah.
Okay, thank you very much.
Okay, are there any questions from our audience?
Okay.
I wanted to,
Yeah.
I wanted to follow up on that
Yes.
question, because you said that it's pretty much impossible to completely eradicate them,
Yeah.
because also invasive species, they tend to just proliferate
Yeah.
regardless of what we do. Would it be, just thinking like in the future, would there be a point where we could potentially be harvesting enough that it's not an invasive issue, but we are still allowing it to
Yes.
proliferate, so the company is still growing, but it's also not an
Yeah.
environmental issue? Is that something that is possible?
Yeah, so it's possible because that would be like the best balance for both like the marine ecosystem, because they also play a huge role in the marine ecosystem. It's just that they are like excess now. So if we are like harvesting 6 months before we take the amount we need, it allows time for the ecosystem to recover, and then we just keep it balanced. But since this project is not really on policy, it will be up to the policy makers to be able to say, okay, this species is no longer a threat to this particular ecosystem or that particular ecosystem. But as it stands of today, these three species are like highly like threats because they're everywhere.
Yeah. Sorry, this is about the last result.
The pH?
Okay.
Yeah.
So you said you integrate this by adding some more acid compounds.
Yeah.
But it's already studied, and I'm wondering whether for the skin specifically you need it to get a pH of 7 or a little more acidic is still okay.
A little more acidic is okay because like the skin has like a mantle, an acidic mantle, and like it does well in the skin does well in an acidic environment. Also because the bacteria that lives on our skin are like pretty acidic loving bacteria, so it would make sense to just keep it within the range that has been given like based on international standards and all that than to exceed the range because it's pretty much pointless since the product, if we were to take this product into production today, it will still have to go through cosmetic regulations and law and they will still do all the tests and if they see the pH is not good enough for like consumer use, it's basically like a waste of time, energy and all that money. So we just keep it within that range, 4.5 to 6.
Okay. Thanks very much.
Yep.
Control L,
Control L.
Everybody, so now we're going to hear from Dorian about diatoms. Are you ready?
Okay, thank you. Hi, my name is Dorian, and my master's thesis was about bilateral quantum towards a multi-product diatom biorefinery. So this presentation will generally focus on, I'll cover the introduction, followed by the experimental design, showing my results, the ongoing work, and the conclusion, because I've yet to submit my manuscript, so I still have some experiments running in the labs. So, microalgae are a diverse group of single-celled organisms who are autotrophic in nature through the process of photosynthesis. They're a source of essential bioactive compounds, which are heavily utilized in different industries. So these industries include biofuels, pharmaceuticals, medicine, food, and aquafeed. And aquafeed was sort of my focus for my thesis as well. So diatoms are a subgroup of microalgae, which are characterized by the presence of fructules, which are the silica cell walls surrounding them. And they contain bioactive compounds such as pigments, polyunsaturated fatty acids, beta-glucans, proteins, among many others. So my species of choice was Phaeodactylum jocundum. And now this is a model diatom species whose genome has been fully sequenced back in 2008. We're making it great for omics studies. And it's actually one of the most researched diatom species right now. One of them. So it's ideal for large-scale cultivation, and it grows rapidly in laboratory environments because of its wide environmental range. Now, it also contains several high-value bioactive compounds, such as fucoxanthin, EPA, which is a polyunsaturated fatty acid, and crisolamin, which is a beta-glucan. And All of these bioactive compounds, they have multiple benefits. All of them contain antioxidant and anti-inflammatory properties. But they each have benefits which would be ideal as an alternative source for the aquaculture industry. So the extraction of these compounds are usually done in biorefineries, and the biorefinery approach is important. A proper approach is very important. Ideally, it would be a zero-waste process, which utilizes multi-product extractions, which will make it economically viable in the end. So one way to approach this is water-based biorefineries, which use water-based methods such as ultrafiltration and enzymatic hydrolysis to extract, separate, and convert the entire biomass into usable products. Now, water-based biorefineries are good because they avoid harmful chemicals, they minimize overall waste, and they produce multiple valuable products in the end. And this is generally achieved through the extraction of lipophilic compounds at the end. So you do your initial extractions, you're left with the residual biomass, and then you can extract more compounds from that biomass, such as pigments, for example. So typical biorefinery process starts with pre-treatment, which includes cell disruption, and this can be achieved through different methods, such as mechanical and non-mechanical. Non-mechanical usually focuses on either chemical hydrolysis or enzymatic hydrolysis. They're the more common methods to use, but there are others as well. Mechanical methods uses shear forces to break up the cells, usually pressure, and so on. After pre-treatment, there is fractionation, so this enables the separation and purification of the different extracts. Again, different methods, laboratory methods are generally smaller scale, and then we have the industrial methods, which are large scale for a lot of volume. And lastly, it's stabilization. So the extract that you extract, in the end, you want it to have a longer shelf life than it initially does. So stabilization is the last one. It's usually very important in this procedure as well. So the objective of my thesis was overall the unit optimization of a water-based biorefinery process, and specifically I focused on the enzymatic hydrolysis to produce a protein hydrolysate for the aquafeed industry, while simultaneously limiting the lipophilic compound degradation, so pigments in my case. So in terms of experimental design, we started off with the dried biomass, which we resuspended inside distilled water at a certain concentration. After that, we followed with cell lysing, so we destructed the cell to break up the cell walls, which is important because it helps with the extraction process. After this, we did a microfiltration of the entire volume, and now it was ready for long-term storage for the following experiments. We did this at the very start of the thesis to have enough biomass for the subsequent experiments to use them. So in terms of the experiments that we've done, it was generally two different types of experiments. So first we focused on the actual enzymatic hydrolysis. After that, we tried to quantify the Foucault-Chanting degradation within the biomass. So we focused on different parameters such as the total biomass concentration, the pH, enzyme to biomass ratios, temperature, and duration of the whole hydrolysis procedure. And the way this was done, the overall idea was to incorporate the enzymes into the biomass. So the enzymes we focused on was alkylase and flavorzyme, and the reason for this is because these two enzymes are relatively robust, so they can be used for different purposes, yeah, they're readily used in the industry already, so they're really easy to source. which is beneficial for us. So we included the enzymes for different experiments in 2 ml epindophs, and this was put into a thermomixer, which was incubating the samples at the parameters that were chosen. After the incubation, we centrifuged the samples, collected the supernatant, and then did the spectrophotometric analysis to quantify at first the degree of hydrolysis. This was done using the OPA method, which looks at the percentage of peptide bonds cleaved in the end. After this, so the initial centrifugation, we were left with the biomass pellets. We took the pellets, resuspended them in ethanol, and did an ethanolic extraction of Fukushima. And then the same procedure, collected supernated, and did a rapid quantification using the spectrophotometric method. Now, after the conditions were optimized, the one thing that we're left with is the experiments on the bioactivities, because we were interested to see how they can contribute to the aquaculture industry in the end. So this is yet to be done, and this will only be done under optimized conditions. And basically, we'll run a multitude of antioxidant assays as well as the anti-inflammatory assays, which are available in the labs that I've been working in. And we also utilize Design Expert in our studio for all things statistics and software wise. Now, to move on to the results. So, in terms of the enzymatic hydrolysis, we looked at the 2 enzymes, but we focused on one at a time at first. We initially focused on alkylase, and it was a step-by-step procedure of optimization. So we looked at a parameter at a time, and then we used the optimized parameters for the following experiments. So we first looked at the biomass concentration along with the pH and enzyme biomass ratio. This we looked at in unison. So these 3 graphs display the degree of hydrolysis from the enzymatic hydrolysis. And what we can see here, so there are 3 different concentrations. We have 20 grams per liter, 30, and 40. Over here, we can see the pH that we tested. We tested from 7 to 9 pH. And on this slide, we can see the enzyme to biomass percentage. Our goal is to use as little enzyme as possible because it's expensive. So from these graphs, we can kind of see that the highest degree of hydrolysis was achieved at lower concentrations. Both higher and lower percentages of enzyme were relatively equal, but at pH 9, we observed the highest degree, which is what we're looking for. And the running hypothesis is that this is likely due to the low viscosity of the volume, so there's more mixing involved, so on. So this is the degree of hydrolysis, and these are this is the Fukushima quantification. So this is how much Fukushima we've been able to extract from the biomass using the previous parameters. Over here we can see that the highest was reached with at 40 grams per liter. But if you look at the individual values, they don't really change much. They're pretty consistent throughout, and there is limited degradation. So after these parameters, we've observed that. So let me just show you. We've observed that it kind of peaks on the sides. So we thought there might be potential to kind of extract more than what we can see here. So we wanted to do a validation experiment, kind of increasing the parameter range. And we did exactly that. A validation of the results, and we came to a conclusion that yes, lower concentrations with low enzyme to biomass ratio do prove to be more effective in the end. And again, the Fukushima difference is not, it's almost insignificant for some areas. So after the validation, we've concluded that the best is 20 grams per liter at 2% biomass to enzyme ratio and at pH 9. And these are the parameters that we used for the subsequent experiments. Next came to temperature dependency. So we looked at a range of different temperatures from 20 degrees Celsius to 40. And to sum it up, 25 was the best temperature. You can see that this showed the highest degree of hydrolysis, well, this showed the highest Fukushima extraction, which was interesting because common practice with alkylase is typically 50 degrees, and it works best at 50 for most things, but it did not for us, which was quite interesting. And also, we didn't want to exceed the temperature because, as you can see here, Fukushima degrades at high temperatures. It's very temperature sensitive. It's a sensitive compound in general. So we wanted to limit this as much as possible. So final consensus, 25 degrees Celsius. After that, we did a time series experiment. So this experimental design was a little bit different. Up until now, we were using 2 ml Eppendorfs and the thermomixer. But this was our first scale-up experiment. Because we needed to monitor the pH and everything else, and until now we were using buffers. So basically we did scale up 3 reactors for a total duration of 4 hours to see at what point we can see an exponential increase in degree of hydrolysis and an exponential decline, or a rapid decline in Fukushima. And we've seen that based on these results, the degree of hydrolysis increases exponentially right at the start, and the Fukushima declines right at the start. This might be an experimental error, because at this point we were using magnetic stirrers and stirring the liquid, and we were keeping it at a certain temperature. So the heat might not have been well distributed, which might have resulted in a rapid increase from when we sampled it. But we came to a conclusion that the ideal would be anywhere between 60 to 90 minutes of enzymatic hydrolysis for the following experiments. So that was the optimized version of alkylase. Now we focus on flavors. It wasn't as long because we had to use some of the previous parameters, because after this we focused on the combination of the two, which based on literature proved higher results throughout. So enzymatic flavorzyme focused on temperature, pH, and enzyme to biomass ratio. The highest was observed at 35, at a higher biomass to enzyme ratio. And in terms of Fukushima extraction, the highest was again observed at 35, but minor differences throughout. Basically, we came to a conclusion that the temperature should be at 35, that's the optimized condition. At 2% biomass, because again, we want to minimize, at 2% enzyme, because we want to minimize enzymes to make it more economically viable, and pH 9. So that was great for us. And this brings me to the dual enzyme treatments. So this was our end goal. We wanted to combine the enzymes, the two optimized versions of the enzymes, and create a final optimized procedure. And let me just explain the graph. So we had 5 different conditions here. This one is just alkylase, followed by just flavorzyme. So this is the degree of hydrolysis table. Alkylase, flavorzyme, this was the combination of the two, and we combined them at the start. Next is a sequential addition of the two enzymes. So this one here, they were both kept at 25 degrees, basically 1 hour alkylase, 1 hour flavorzyme. This one here was again, 1 hour alkylase, but before we added the flavorzyme, we increased the temperature to 35 degrees as per the optimized conditions. And as you can see, that showed the best results, actually. And there is an insignificant decrease in Fukushima extraction as well, so great for us. So that was our optimized version of the results, and what's left to do is the bioactivities and to see how they fare in terms of that. And to conclude everything, we found out that it is possible to optimize process of protein hydrolysate production with overall minimized Fukushima degradation, and this can be achieved through the sequential addition of both the enzymes. And also, it was interesting to note that our optimized conditions, the results that we gained, they match the results that we found in literature of the best hydrolysis possible for these types of enzymes. Thank you for listening.
it was very nice because of the icons, figures, really nice. I really enjoyed it. I'm not an expert on this field, so my questions are just curiosity. I think the initial hits the dry one, that's confused for the extraction. Is a mixture of species or are just selected ones?
So we only focused on one species, which is Phaidactyl utricornicum.
Okay, sorry.
And it was the dry biomass. Sorry. No worries. So it's only one species that we focus on, Phaidactyl utricornicum. It's the readily used one. And we had it in dry, we had the dry biomass, so we had to resuscitate that.
Yeah, of course. And how long does it take this extraction?
You mean the
procedure of the extraction when you set with temperatures and stuff? So to obtain kind of product then.
So are you including the pre-treatment and?
Yeah, all this procedure. How long can
it varies. It depends on the products you want to extract. But so in terms of pigments, for example, the stabilization methods that you use or that we've used are so we use a freeze dryer to dry up the pigments so that you have them in powder form. Then you can utilize them in different methods. And the freeze drying process generally takes quite a long time, especially for bigger volumes. So what I wanted to say, it really depends on what you're trying to extract. Sometimes it takes a relatively short time, other times longer. You're looking at 1 week plus per extraction. The volume is also a big factor. If laboratory scale, it's relatively fast, but when you want to do it commercially, unless you're using bigger equipment, more powerful equipment, it's going to take a really long time.
And the reactors, how are they designed? So, like these reactors that you use, you said that at some point you used these reactors in which.
So those were essentially just. No, so it wasn't a reactor. It was just for the experiment. We used a magnetic stirrer inside a beaker. So the beaker was the reactor. Yeah, so it's like I say large scale, but it was larger than the previous ones that we've used. So this was our first larger-ish experiment that we're doing. No, it's a valid question.
Thank you, Dorian, for your very nice presentation. Well, actually, I'm also working with data, so I'm very interested in your work. And I have a couple of questions. So you talk about the biomass, the dry biomass, but do you also cultivate the data for the extraction?
So we didn't cultivate the biomass specifically because this thesis was a part of a larger ongoing project with different companies in Portugal. So I've done this at CCMAR in Portugal. It was part of that, and basically the other companies that provided biomass for us. The labs that we've used, they do have a culture lab per se, but we didn't use those experts from that.
So you don't have any idea of which kind of light or which kind of...
Those parameters I'm really not sure about.
And what about the cost? So you said that the cost of the enzyme is high, and so the best combination you presented is a combination of 2 enzymes.
Yes.
So do you think that the products you will take will cover the cost of the 2 enzymes, or you propose a biorefinery with 2 products majorly, so proteins for activity and sanity? And do you think that selling these products can cover the cost of adding 2 enzymes for the extraction?
Definitely. I mean, economically, the entire process needs to be looked at in terms of the cost. What I've been doing is just small-scale experiments compared to commercial volumes. So as you increase the volumes, as you increase production, you're going to look at higher expenses, basically. and we just wanted to minimize this as much as possible. These are highly desirable compounds that we're dealing with as well. So there is definitely some application, and you can definitely recover the costs that you're spending. But in terms of the actual costs, I don't have that data. No, it's a very profitable industry.
Okay, thank you very much for your presentation. I really enjoyed that. It can be easily followed, it was made very well, addressing the public, pointing to the relevant information at each slide. Thank you for that. Thank you for that because I'm not familiar with the topic. It's all far beyond my expert. Thank you. And I want to stop a little bit on the results. Sure. It showed these 3D graphs, which are not.
This?
The first one, please. Yeah, one more. That. So these are not easy to explain. You don't have to stop there and explain the beginning of the taxes and so on. But here you have that they are relatively flat. So probably there's not a lot of effects in between the variables.
The middle areas. So the reason for this, the experimental design, the experimental design that we bought for this was a box banking design. So basically, how that works, it looks at the middle values the most. So you're having more replicates in the middle, which ends up producing these sorts of graphs. It would be better to increase the experimental design to focus on more samples, but we were quite limited with the time, so we had to kind of combine them.
And do you think that also explains why you have two maxima there in the third graph, two range parts?
Is that the which two?
Would you explain that?
The way I see this is that it probably does not dip in the middle as much, and that it's probably relatively uniform throughout.
Probably need to redefine your color scheme.
Yeah. But as you can actually see, just purely based on the values, they don't change much. This is just an internal difference between them saying that these are the lowest and these are the highest, but actual quantification, it's relatively small.
And you can keep the same color scale with the same values for all the labs.
Yeah.
So in that one, probably you see everything like green or yellow.
Yeah.
What I mean.
So these were all done using Design Expert, the software that I mentioned earlier. And it's a software heavily used for optimization experiments. So, for example, the red dots are my outliers in the experiment that I designed. So this creates a little panel based on potential optimized results, based on potential optimized parameters based on your results that you've tested.
I have a question about the temperature dependence graphs. You mentioned that you find there that you find that the optimum temperature was yes, it's 25 degrees.
Yes.
But I think I understood that the best temperature in literature was 50.
So
50.
So 50, 50. So 50 was is considered common practice for algorithms. It's the literature that I've explored, that we've seen, they always use 50 as a standard. There are several reasons for this. So each data species requires its own optimized conditions. And this is largely due to the cell walls, because they are all different from one another. Some require higher pressure to break up, others require less. It's each species is independent, basically. So 50 kind of became the standard for industry. But we were also expecting higher values, higher temperatures. So that's why we were surprised when we saw 25. It means the antigen is actually working at that temperature, so that's great for us. You don't have to spend so much on the overall heating of the bioreactor, so it cuts down costs. So the reason we didn't try, because as you can see here, Fukushima degrades rapidly at higher temperatures. So we wanted to limit Fukushima degradation, and we kind of stopped at 40 because we can see a rapid drop. People say so. The cell wall is actually the biggest enemy.
Okay, thank you very much.
Thank you very much.
Any questions from our audience? Yeah?
Let me use this presentation. I'd like to know what are the activities that you will do in the
So we will primarily focus on antioxidant and anti-inflammatory activities, and again, these are just the ones that are available in the labs. I still need to explore this area, but it's going to be a comparison between different benchmarks of other species that were used in vivo and in vitro. So we'll kind of validate our own optimized conditions and see if they have, or how much of a potential they have in the aquaculture industry.
Thank you, Lori.
Thank you.
This concludes our session. Congratulations to all the speakers. We have our lunch break now. We'll turn it back. Thank you.